Hans on part of the organizing committee for the National Agricultural Producers Data Cooperative. So just as a reminder, exactly what that is the overall goal of the project is to develop a blueprint for a national data framework and cooperative where producers, universities, and not-for-profit entities can store and share data and develop powerful tools that enable producers to maximize their production and profitability. So, that's a mouthful, but just to get a refresher, kind of who we are, and as part of this series of projects that we have this webinar series. So we're fortunate enough today to have João Dereia, hopefully I pronounced that somewhat close, from the University of Wisconsin Department of Animal Dairy Sciences and Biological Systems Engineering. And he'll be talking about leveraging artificial intelligence to optimize farm management decisions. So again, we'll have about 45 minutes for the webinar, and then we'll open it up to questions on that. So please feel free to drop those in the chat, or we can raise your hand on the Zoom icon, and we can get to those at the end. So I'll turn it over to Dereia. Thank you.
Thank you very much. Let me share my... Okay, can you see this well?
Yep.
Okay, there you go. Thank you very much. Thank you, you all for attending this presentation. Thank you, Jennifer and Trenton, for the invitation to present. So today I will talk a little bit about the research that we are conducting in our lab. I think there's a topic that is interest for everybody working with data in agriculture, and in my case, I specifically work with livestock species. So I work with animal farming. I'm using a lot of the technology to collect animal level information for management decisions, and I collaborate a lot with geneticists that are interested in phenotyping, precision phenotyping, and how this could help them for genetic selection and animal breeding programs. So for today, what I will do is I will quickly talk about the importance of precision measurement and of target traits in animals in different conditions, and how we can use technology that could measure these traits in space and time, and how this can generate novel traits, or how can this be used for real-time monitoring. And I will give some examples from our lab about the use of this technology for growth development and animal health identification, and for feed analysis too. And we will close with a case study. This is a project, it's a long-term project that we have, that is we are monitoring animals throughout the lactation, so from day 0 of life up to the end of the first lactation. And we have some preliminary data from that project that I'll present today too. Okay, so why we are interested in sensing technology for animal, for livestock. We are able right now to collect and integrate data from specific source, and that could be weather, crop, water, soil, economics, or price of commodities. But it's been really difficult in the livestock side to collect animal level information. And it's difficult because sensing technology developed to collect these 24/7 is not that available, or is not that easy to implement, right? So that's why there is this huge interest in data collection or in phenotyping at the animal level, is to deploy this device that could vary from microphones, camera, wearable sensors, that could be able to give us animal behavior, traceability, body composition, methane emissions, and with that information, we could then optimize farm management decisions, or we could also make a lot of progress in terms of genetics. And so there are then two important points that by having that data, we can achieve. One is what I just said, farm management decisions. And what is important to think about here is that these farm management decisions create value on the sensing technology very quick for most of these applications, right? So if you have a sensor that can track animal health and improve health of the herd, then we would gain, or we would create economic value very quick, right? So the animals would want to get sick. You don't need to treat the animals. Milk production probably would increase, or average daily gain would improve. So that return on the sensor investment, it's shorter than in the case of, for example, genetic selection. In the case, specifically, I'm talking about cattle or dairy cattle. If we collect some phenotypes, we take some time for the progress of the genetics to come back and contribute with that. So the reason I'm talking about these two short and long term we're using dairy cattle as an example here, is that this alignment of which sensing technology we are deploying that will help farm management decisions and will generate phenotypes that are important for genetics as well for animal breeding programs. So from my collaboration with geneticists, I've seen that these synergy, it's very important if we want to collect data very quick and in large scale that will benefit both sides. So it is from my collaborations, I noticed that it's going very well combined. And on top of all of these, labor is a problem, and in all sectors, not only in agriculture now. And so having people to take notes or to control inventories or to observe animals at the farm, this is definitely a challenge. And so sensing technology can help that a lot. So I'm very interested in different technologies, electronic feeding aids or collars for behavior, wearable sensors, automatic milk feeders, spectroscopy, again, wearable sensors. So we have published using all different type of technologies to monitor different phenotypes. And in all these papers, we are trying to do something related to management or detect disease, early detect disease, or predict dry matter intake, or estrus detection, or grazing activities. But the reason I'm presenting all these titles of these papers is that although I'm very interested in that, and it just pretty much to prove that I'm interested in all the sensors, but today I will dedicate most of the presentation for computer vision, for image analysis, how we are using these to generate these phenotypes, and how we are using these to track animals at the farm. All the investment we are making to equip the farms and have these running in a more large scale. Okay, so this next slide is just to prove or try to convince you why we are investing so much money and time and effort and training from students in that particular area. And one thing that really bring a point that it brings always to our attention is that the value of the data when we work with computer vision or images specifically compared to other sensing technologies. Right? All of them are important, and again, we are using all of them. But this here just to give you a sense of why we are dedicating more time to computer vision. So we have these two graphs. They are generated from an accelerometer, a 3x accelerometer. So we have here X, Y, and ZX plotted. And we have two distinct behavior 1, behavior 2 in graph 1 and graph 2 here, the top and the bottom graph. And we can clearly see the signal is different from the motion that the animal is performing over time. And if I ask you, what can we do with this, or how can we use that? And the answer is probably we cannot do much because we don't know what the animal is doing, or we need label. Then if I tell you, okay, if I give you information now that this is grazing, this is non-grazing, and then we may say, okay, now I can train a model that can use this as input and predict now the animals grazing activity, non-grazing activities. And that is in fact what we did in this paper. But then this brings our first point that if I don't have annotation, if I don't have a label for these data points, I cannot do much with that. Right, and which is a little different when you think about image, because if I give you a segment of video like that, you have animals that are grazing or animals that are non-grazing, and you can definitely see you could possibly annotate information here, right, from that video segment, and you can even see more. You can see there's not only grazing, but you can see that it's grazing, it's given a step, number of bites per minute of the animal. You could see probably the type of pasture. The pasture is green or not green, or so on. The animal is standing lines and all that aspect. And this is going most of the time beyond the primary interest that we have when you start collecting the data. And so over time, if you want to go back to the data and apply other techniques to that, you could definitely extract other phenotypes that were not taught initially. Okay, and so here's just another example of that. You look to a picture like this and you see the number of animals. You can count animals, potentially predict the breed of animal type, and if it's raining or not, and so on. Again, just to reinforce that how valuable is the information that we can have in a single image. Okay, and again, this is the reason we are collecting more and more data, because we are leveraging that over time, especially as machine learning algorithms are developed in computer vision and making possible things that are at this point they were not. So one example I will start showing is related to animal identification. And the reason of this animal identification problem that we are trying to solve is because you want to track animals for disease or metabolic disorder, health problems during transition period. And you'll touch these, what it is pretty quick. So in our farm, we have about 500 cows and lactating cows. And these are the exits of the milking parlor. So every day, the cow will walk through this lane twice a day. It will have two lanes here, one and the other one here, as you can see. Maybe I should use a laser point. One here and another here. And we have these 3D cameras, these depth cameras, position top-down view in one of the lanes, and another camera here, position top-down view in another lane. Right. So we have 2 lanes in one side, 2 lanes on the other side, so we have a total of 4 lanes. And these cameras can collect RGB image, regular pictures, as we use take from our phone. We have an infrared image like this grayscale image you can see here, and we have this depth image that it maps the distance from the camera to the object. So here, as you can see, is dark blue. This will have a value indicating that the distance from the camera to that dark blue is closer, and if it's dark green, the number will indicate a longer distance. So you map the distance from here to the object. So then we can have a 3D shape of objects using this type of device. And so one way that we are using these, and it's automating the process when these animals go to milking parlor. When they leave, we acquire a sequence of frames from these animals. So basically, this camera monitor every single second the distance from what is located to the floor. And so if the distance change, it means the cow is walking through the lane, right? So because then it's not the real distance. Let's say this is 3 meters. If the cow is walking now, the camera is mapping the back of the cow and the distance is changing. So we acquire a sequence of frames. And because the animal is walking, we can collect good image, bad image. And so the first thing we developed was we need to run an algorithm for image classification that would classify images that are good from images that are bad. So we had this label of good. Good is all the image that we have the entire cow in the image, and bad is everything else. No cow, cow that is cropped and blurry, and so on. So with that, we reduce a lot the data that is collected through the system. So for example, here in that given example, 52,000 images, and then we narrow down to 19,000 images. Also, there was a lot of like bad image. The animal was not perfect in the middle, so we didn't want that image. And the reason that we want to put this filter is that all that data is going to the cloud. So whenever the image is collected, we send this to a blob in the cloud, and then all the pipeline of automation for calling the models to perform analysis is done. So we don't want to send images out there that are not good. So if the image is good, then we call image segmenter. So here you use a network called UNet. Here's exception. I can go in detail about these deep neural networks that we use for each task. I will go quick here just now to explain first the pipeline. And so if the image is good, we then segment the body and remove all the background. And if the background is removed, then we have this masked image for infrared and for depth image. So we can, for example, perform animal identification in that image, or we could run image classification, or sorry, body condition score in that depth image, or we could compute body volume, body weight, and so on and so forth. The interesting aspect of that pipeline is that we automate the collection with not touching the animals, restraining the animals, and then we have the data ready, and you could evaluate 500 cows every single day in terms of body condition score, identification, and other traits that we are interested. So the next question everybody asks is how you identify animals. if we're using RFID or not. Definitely RFID is the most used technology for animal identification. We are trying to run systems that we only use computer vision. And so in that case, we are testing how accurate is the system to identify animals based on the black and white coat color pattern, right? Like here. So this works like some sort of QR code or barcode. And so we are achieving very good accuracy to identify this group of animals based on the black and white coat color. The problem is, as you increase the size of this classifier, or the number of animals, in other words, we have more black animals, complete black animals, or complete white animals, or solid homogeneous coat color. And then those won't be able to recognize them very well based on the color. So what we tried them was to develop a models that would receive as input 3D shape. And these could be done using depth image as input, because each pixel information is related to the distance from the camera. So basically maps the surface of the body of the animal, the back of the animal. Or we could use 3D representations like voxels or point clouds. So that's what we did here. We test these three ways of receiving an image or training a model, receiving only depth image or only maps with no color information, just the surface, the back surface of the animal. And then we achieve very good accuracy, or here we report an F1 score of 0.8. And one interesting comment that I have here is that we perform this training and validation using random or chronological split from the frames that we obtain of these animals. And as we would imagine, if you split this randomly, you have frames that are very close in your training testing set. So values are inflated, as you can see here, with F1 score of 0.95. And then when you break that validation and training set chronologically, then frames that are close in time, they remain the training set comparing to the ones in the testing set. So it's normal to see that reduction on the performance of these models. And so the other question was how this works temporally. If I train a model with animals with the body surface or the back surface today, and the animal is, let's say, is a dairy calf, and this animal is growing, so in 5-6 weeks, can I still identify this animal? And so we start doing this. So we skip 1 or 2 or 3 weeks, and then we train the model with early image from the early stage of development, let's say week 0, week 1, and then start skipping these weeks. And so the model performance is still good. And so in this short term of 6 weeks, which was this trial, we said, yes, if you train with week 1 and you go to week 6, which the animal is very different already, is growing very fast, we can still recognize the animal based on the surface only. So now we have a grant, which we are looking into 2 years development and see if we can recognize the animals very late stage, like 1 year later, 2 years later. And if not, how frequent do we need to retrain the models in order to be accurate for animal identification over time? So as part of this problem, we are in the farm system that I showed you, we are developing these because we want to detect disorder during the transition period. And these disorders are related to body tissue mobilization. So we are looking to the shape of these cows. That's why we collect this 3D image, this depth image, and you want to early detect health or metabolic disorder that happen because of pregnancy or because the cow is mobilizing too much energy to supply milk production. And this is relevant for dairy systems because 67% of most of the disease or the metabolic disorders happen during this transition period. And it's 3 weeks before calving and can go up to 3 weeks after calving. And so basically, if you're not familiar with that period, what happened is the cow is producing a lot of milk, but the cow is not eating enough to supply the demand, the energy demand. So the cow starts mobilizing body fat, body reserve, and so if depending on the severity of the body tissue mobilization, the cow can get problems as ketosis, hypocalcemia, retaining placenta, metritis, endometritis, and so on. And so what you want to do with that, it's monitor these cows before calving, after calving, and making sure this image are informative enough, or not making sure, but use this information containing this death image to build models that could help us early detect animals if that severity of the mobilization is too high. So the way that's done now, body condition score is usually visual assessment, is subjective. You may have a 2.5 cow and a 3 cow, and someone may not be in agreement with between two evaluators, for example. And another interesting thing is that there are changes in the body shape that are just hard to visualize with human eyes, let's put it this way, right? So we have this cow here, 21 days before calving, and the same cow 14 days before calving. 3 evaluators gave body condition score 4. Another 3 evaluators gave body condition score 4. And then when we took the death image of these cows and we overlaid that, we saw different in shape. Some of the cows were already shrinking or changing the shape. And so it's really hard to pick up this difference. So that was another reason that we decided to look to these death image and try to monitor them over time in order to classify body condition score and disease. So we ran this trial with 76 cows. So we monitored these cows 21 days, 14 and 7 days before calving. And we took blood samples pretty much every other day. And we analyzed blood BHB and plasma NIFA concentration. And we took, for each time point before calving, we took a total of 27,300,000 images. These are top-down images from these 3 time points. And so what you want to do then here is using these 3 time points and predict an event of subclinical ketose that could happen up to 14 days after calving. So this is already a very complicated problem because my last data point is 7 days before calving. And if I have a ketosis case, the day 1, then I have almost a week of gap between my last data point and my healthy event that I'm trying to predict. And this can really get worse if my healthy event happened at day 10 or day 7, right? So it's even bigger, the gap. So here we are not treating these images as series because we have 3 time points only. And so the project we are conducting now, we have 60 time points before calving. So we have 60 days. So then we're going to start treating these as a time series analysis or data when we process these images. Right now, we treat them as independent predictors. Okay? And so what we are doing is we are extracting biological features. We call these biological because we understand what they are. So for example, the body volume, the surface area of the body, and so on. And we are also extracting convolution neural network based features. We are using an architecture that was trained previously in other data sets to classify body condition score. And we are using this architecture to extract the last layer only of this convolution neural network, which is composed by 1,024 features. And so basically the convolution process, we extract features initially in the top feature maps, very general, related to the edge, contour, texture, and then these become very specific related to the object we are training as we go down on these layers of convolution. And lastly, we have these 1,024 features that would be used to classify body condition score, which is not the case here because we are not classifying body condition score. We just interest on these features, hopefully related to body shape. And then we are combining them with the biological features and use this to predict subclinical ketose. So here is just some of the gradients from the activation maps that we obtain from the network, and basically looking where the network is putting attention as we classify different body condition scores. So that's more related to the body condition score. So the results we achieved with that was we use these features in 2 analytical strategies. So I won't be discussing this in detail, but I'll be happy to answer questions later and discuss. So we usually use a partial least squares discriminant analysis, and we also use gradient boosting decision tree. And we achieve better results with partial least squares. And so we have F1 score of 0.75, recall 0.91, precision of 0.65. So it was better than using the body condition score as a feature only. So extracting all these features that I just described. And so What these numbers really mean is that from 65% of detected cows actually got sick, and 91% of the sick cows were early detected. So this was very interesting for us because although we have 35% of misclassification here that are basically cows that did not get sick, and the model said this cow got sick. So we have 35% of this here. But this it's less critical than the model letting cows go sick, because this is a much more expensive problem to deal with than these false positives, which would increase only cows being checked more frequent than it should. I'm not saying this is what we want, but what I want to say is these were basically based on 3 time points of death image. I didn't include any information related to genetics, diet, previous records and other lactations and all these. I didn't include that. We didn't include at all. So we were very happy with that result, because these only took these 3 time points of these images. And so now what we're doing is including more time points and more tabular data to complement what the image is bringing. So related to shape as well, we have done a lot of body weight prediction in pigs, in beef cattle, and other groups are doing these as well. And the results are very good from our group and from other groups. If we go in the literature, we have a lot of that that would prove what I'm saying. And here, for example, it's one of the trials. We predict animals at weaning, stock phase, beginning of feedlot, and end of feedlot. This is beef cattle. And what we were interested after that was, can we predict a meat cuts with the animal, with live animals, with death image of live animals? And that's what we did in this particular paper here. We use ultrasound image, we collect the ribeye area and circularity with a live animal, and then we collect a death image, a top-down view, as you can see here. And then we try to predict using this top-down view image, this death image that would map the shape of the animal body. We try to predict what the ribeye circularity and area should be. Okay. And so to do that, we basically train a network. The way we extract the features to make these predictions of the ribeye area circularity was by training a network that would segment the animal body. The only task here is segmenting animal body. So we use death image and we train these different networks to segment the body of the animals. And we use the feature maps that we learn during training as a predictor of the body of the ribeye circularity in area. So we use different approaches and we tested if a particular segment would perform better than other. But we also compare these with using body volume, body area, and biometric features. So overall, they perform similar, very good accuracy and precision to predict ribeye area and circularity. As you can see here, R-square 0.74, mean absolute error of 7%. But then when we use biometric features, and here they were body volume, let me see if I have here. No, they're not here, but they're in the paper. We use body volume, body area, circularity, max length. So biometric measurements that we would obtain from the animal body, and they were able to predict as good as what the deep neural network for segmentation were learning from the body shape. So that was a very, very intriguing and very interesting study that could open the possibilities for precise nutrition. We could group animals better and do other things related to management. So we are very interested in behavior as well. We have used group housed animals to monitor individual behaviors, and these are basically, in that particular case here, that's based on animal identification. So here we are mapping animals when they cross the head on the headlock. So if the animal is coming to eat, we compute the total time the animal is eating, the visit duration, the interval between visits, the total number of visits. And so the idea here is that we can monitor feeding behavior. So if animals are not going to eat, we can see that drop very clearly. And so we are very interested on the behavior and feeding behavior monitoring as well. And in a more large scale here, we have cows. We have a group of 60 cows, and we are monitoring the entire group. And we are monitoring if the animal is eating, lying, standing, drinking, and we can map the animal in the barn, which is it's very useful, and it can give us the chance to look into social interaction based on distance among these individuals and study their behavior and social interaction more or more precisely. And related to feed particle and feeding behavior, we are not only looking to the animal aspect, how much time the animal spend eating or drinking. We are also very interested in using image technology to map or to characterize the feed that the animal is consuming. And so there are enough papers and there is enough literature showing how important feed particle size is to regulate dry matter intake, to affect milk yield and milk composition and health. The big question always is how we measure feed particle size in an efficient way. The methods that are the gold standard methods that take time for us to monitor, to measure, right? So usually is a shaker box. You put this feed stuff there and you shake, and then you measure how much was retained in each particle on each screen. And based on that distribution, we can assess how the diet is being mixed and how this would affect performance of these animals. Okay. So what is interesting about that? It's what I'm just saying about feed particle distribution. They are more physical characteristics of the feed. We are automating a lot the chemical part, the chemical composition, right? So we have used NIR, near-infrared spectroscopy, to predict compounds and feed stuff. This is being used in research, in industry. So we can go with a handheld NIR, we can go with a bench NIR, and we can predict fiber, starch, digestibility, all these things. Recently, this has been used a lot for animal phenotypes. But one interesting aspect of the NIR or this automation, this is more on the chemical composition, as I mentioned. For the physical characteristics, like particle size and distribution and on, we don't have a lot done, right? So that was some of our interest. And very, very quick here, if you never heard about hyperspectral imaging or the reason we are interested in that, I'll briefly explain. When we have a sample, let's say a corn ground or a soybean or any feed stuff we want to analyze using NIR, we put in this plate and then we shed light into this sample and we generate a spectral composition, a signature for that particular sample. And these will have absorbance or reflectance of the light in different wave length regions of the spectrum. And we use these 1D signals or these absorbance values for all these wavelengths, and we collect thousands of these feed samples, and we train models that are able to, based on a line like this, a signal like that, predict a new sample. If you bring a new soybean, you generate a signal like that, and you can predict, for example, crude protein in that particular sample. Okay. Forget about the NIR part now and think about image. So in image, we have pixel information that will tell us the intensity of a given color. So if we have a grayscale image, we're going to have, and let's say this image is an 8-bit image, so we're going to have 256 levels or pixel intensities varying from black to white, right? And so if we have an RGB image, now each pixel location, each pixel value, we have 3 values, and these values will be the intensity or that can vary. If this is an 8-bit image or channel, these will go from 0 to 255 red, green, and blue pixel intensity for each of these colors that are in the region of the visible light. So for each pixel, you have 3 numbers then. So in hyperspectral imaging, you have much more than that, because usually you have NIR region being added to the image too. So for each given pixel location, we may have 200 values, and these values are the absorbance values for that given pixel location, which is related to the object in the image, right? So that could be a piece of leaf, a piece of stem, each that would have its own spectral signature, and then the image then can tell us a little bit about the composition of the objects that are present in that given image, right? So for example, if I take a picture here, instead of having 3 channels in the RGB, I have 242 channels, and for each pixel location, I have 242 values that would tell me what's the absorbance or reflectance for that given pixel location in the image, right? So this is no different from hyperspectral imaging for crops. The only difference that instead of flying in 1 hectare, 2 hectares, or in large areas, now we are scanning very small objects. In this case, we are scanning animal diet. So here is just a pilot study we ran with 10 diets, which we sampled we got 2 samples of 200 grams from each diet. And we scanned these in a hyperspectral camera with 224 bands. And we also measured the particle distribution using the gold standard, which is the Penn State particle separator. Okay, so we use 2 analytical methods here to understand how particles are distributed in this sample, in this feed sample. And the first strategy, we used k-means clustering based on the spectral signature of each pixel, and after that we extract features from each cluster. Okay, so we fixed k as is equal to the number of ingredients. So if I have 5 ingredients in the diet or 8 ingredients in the diet, then I have k equal 8 or k equal 5, which is an assumption, and not necessarily k or the clusters will clearly or exactly represent each ingredient. But that's the way we start building these. So then the features were extracted from the segmented areas. So for example, we obtained the spectral data from this diet, and we would then get, let's say, this green label here. This is, let's say, k equal 1. This is a k equal 1 of the ingredients. That could be the first cluster. And then we got the binary image related to this cluster, and that is the binary image. And then we compared the average area, circularity, perimeter, all these features from that particular cluster that potentially will represent, let's say, corn leaves. Okay, and then we did this for all clusters, and we extract all these features for each given cluster, and then we use the input as in a lasso regression or partial least squares to predict the amount retained, the percentage retained in each screen. That was one strategy. The second strategy was we scanned this sample, we got the hyperspectral data image from this diet, and then we broke this big image in patches in a much smaller size. So here, for example, we broke these in 32 patches of 256 by 256 by 224 because we had 224 bands. And then we use autoencoders to reconstruct the patches, and we use the bottleneck of the autoencoder as a way to reduce the dimension and get a feature space to predict the amount retained in each screen, assuming that the autoencoder will learn about these clusters as well, similarly to the first approach. And then we use these features in lasso regression, partial squares, and as we did before, and random forest. So the result we found initially of this small trial was the spectral clustering work a little better than the autoencoders. We achieved R square and root mean square that were interesting for that application. For average particle size, the amount retained, the percentage retained in the bottom of the screen, lower, middle, and upper. We can see here that we couldn't predict well the lower screen, but we could predict the other 3 reasonably well for that particular in small pilot, which we are now digging more and studying more how we can get that better, improve, and collect more samples and move on. But this is just an example of how that spatial analysis for the diets and using hyperspectral cameras could help us in terms of that formulation and mixing and management at the farm, and which is for these is extremely difficult problem. Well, not that extremely difficult problem. It's difficult. There is a particular difficult here that is labeling these. We don't have gold standard, or it's hard to have gold standard because we cannot separate well the feed ingredients that are mixed in the TMR. So that's why both approaches we are using unsupervised learning to extract the features, because it's difficult to have data annotated or labeled for that type of sample. And so lastly, what I want to show you, it's the grants that we have for strategic heifer replacement. In this project, our interest is how we can early detect animals that will be productive and efficient, and considering that there is a high cost to raise animals. And so if the animal is not um, a top animal later on in life, and we don't know that, then this would not be great. So how we can use that, and sort of using PTA only, we could account for more for the phenotypes and that will be happening during the life of the animal, and these phenotypes would inform about the environmental effect, and these would contribute also to improve the prediction. So not only using the genomic information, but also combining genomic information and the phenotypes. So in this study, we are following 200 animals from birth to the end of the first lactation. And we have these animals in a high-low plan of nutrition just to create enough variation in terms of growth. And we are monitoring the mammary gland development of these animals using ultrasound, and we are biopsying these animals as well very frequently. And we are collecting blood samples and other measurements to follow the growth development of this animal. So for example, for biopsies here, we have a lot of time points that we are interested to pursue and see what they tell us. And this is run in one of our facilities. We have 30 devices collecting data every single day. We have 30 single board GPUs, and each single board GPU is connected to one of the cameras. So we have 30 GPUs and 30 cameras, one installed in each of these white box, for one in each pen. And we collect a lot of data, as you can see here, a lot for us at least, for what we need to keep and transfer. And this works as some sort of edge computing system. We have one server controlling all these nodes in each pen and coordinating data collection and acquisition and sending data out and so on. And one first thing we are testing here, and this again, as I mentioned before, is a case study, just some of the preliminary data we have. We have, in 10 animals, we evaluated if we could detect the animals 1 year later. And so, for example, we have these animals, images collected from week 1 to 4. The animals were 42 kilograms. And then we collect data from these animals when they were 6 weeks old, and they were weighing 458 kilograms, so 400 more, almost, kilograms. And so these are the differences on the animal. This one example of the animal that were in the data set. Was in the data set. So here you have this body segmented. So it's interesting to see that some black spots, they are here when the animal is small. As the animals start growing, this black spot come up to the back of the animal. And so you're questioning if the algorithm would be robust enough to still detecting the animal well. And so far for this small group, the results are promising. We want to add more animals and do more work for sure. By adding depth images, or not only code color, not only RGB images, and try to create these long-term animal identification based on the computer vision system. So we are also here trying to predict age at puberty of these animals. So we use for that 124 animals from the total that we have of the 200. And we collect image between week 1 and 6 of life. And we try to predict the age of puberty if the animal would be in puberty before or after 334 days, okay? So we are here in this project, and just to give you a timeline, that for that particular age at puberty, we collect image week 6, and we're trying to predict here 300 days later. So to do that, we use these depth images from these animals. So we collect body volume, length, eccentricity, circularity at the first weeks of age. And then we train a binary classifier that would give us information if it would predict if the animal will be in puberty at 335 days or not. Okay, and then we use a live on out cross-validation for that. The results are not great yet. We have a precision of 0.56, F1 score of 0.67, recall 0.84. And they are not great in our opinion for some reasons. One is we have the 1 to 6 weeks of life and try to predict a variable at 42 weeks later without accounting to any environmental effect in terms of diet, nutrition, anything, just based on the biometrics of the first week of life. And so we are adding more images, and we have genomics for these animals. We'll be using that information as well, and nutrition and weather and other strategy to extract features from these images that is not exploring yet. So these are things that we will be trying to accomplish in this grant. And as I mentioned, part of this is related to mammary gland development. So we are tracking how the mammary gland is developing. Some animals will have maybe high genetic potential to produce milk, but something may happen in terms of heat stress or other stress, and this may affect mammary gland development, and then this will compromise milk production. So we are study how the gland is developing under different management and how the environment is affecting that development. And so we're using a lot of image analysis to extract these phenotypes. Here's just one example of data augmentation. So the ultrasound image there, we cannot collect any high frequency. So it's tough to have large data sets that are annotated for things that you want. So here, for example, we have the parenchyma tissue. We want to understand how the parenchyma is growing. So we had initially 396 images, and we then use an augmentation strategy to bring this up to 15,000 images. And so for that particular augmentation strategy, we use polar transformation, basically to move the position of the parenchyma from one side to the other or down to the center, as you can see here. So with these, we had to convert from the Cartesian plane to the polar plane to the Cartesian plane, and then we would move the parenchyma tissue and then go back to the polar plane. So this would bring the image similar to how that was from the ultrasound. So if you just crop the parenchyma tissue and paste here, the parenchyma will be in a position that doesn't look as real as the transformation we're applying. So we compare augmenting the data set using rotation, crop, and other regular augmentation strategies. As you can see here, our regular, but using the polar transformation definitely help a lot training the model and performing the segmentation parenchyma tissues of real images. Okay. Here's just a comment to say that we had people annotating images well and not well. And so definitely the quality of the segmentation affect the training process. And so for images like ultrasound with soft tissue needed to be annotated, an expert make a huge difference in the quality of the model. So those are the things that you need to be very careful as you ask someone to annotate. And we evaluate some of these here. And I think that's my last slide. One thing we are interested in, we are working right now, it's other shape. We are correlating the 3D shape of these others collected with the depth cameras with internal tissue, like here is mammary gland cistern, and also evaluating the disposition and the shape of the other as a phenotype and how this is related to robotic milking systems and other things like that. Well, as final considerations, and I'll be glad to discuss more. I think all this technology and AI, especially computer vision here, can bring a lot of possibilities in terms of novel traits. We can track animals over time. Most of this is non-invasive, as I try to stress and show, and may result in not a lot of stress for the animal. And these can be monitored closer to production conditions, which I think it's good. Can generate cheaper phenotypes, particularly computer vision has a great potential for that. And I work with farm management. I collaborate a lot with animal breeding programs and geneticists, and I think there is a lot of synergies and opportunities in that intersection. And these, of course, it's been very interesting and important in terms of training, education, how we train animal science students, or how we attract people to work in that very interdisciplinary area, right? And I want to acknowledge the funding that I have from USDA, their Innovation Hub, WRF, and all the partners and supporters from my research program, and all these people that actually make all this research happen. With that, I'll be happy to take questions and discuss more with you. Thank you.
Great. Thanks a lot, João. Very interesting. So looks like we had a few questions in the chat, but I'll open it up to anyone else. Just go ahead and raise your hand. I'll try and identify you. But I guess we can go to the first one in the chat. Was from Huang. It says, if some cows are changing shapes, can have similar body or similar coat color, is it not better to use some sort of tagging, i.e. air tag which will give you precise location identification.
Yes, that's a very good point. I think all these other technologies could definitely be used and should. In our particular case, what we are trying to do is trying to develop a system that we will not need to combine technologies, because just we have seen that as you add more technology one on top of the other, they need to communicate, they need to work well, and it's more sensors to be deployed. I think for the point that we need to combine, but what we're trying to develop in terms of research is to try to not combine, to have a system that could be unique in terms of monitoring the animals, in terms of phenotypes and traits, and at the same time recognize the animal. But I agree with you. I think these tags could definitely help, including help to annotate some of the data that could be used by the imaging system.
Great. Thank you. Jennifer, do you want to ask your question? Or you might have more details on that.
Oh, sure. It was the point about one of your research questions. You were collecting images over time. I think it was like 3 different time points. And you said that for the purposes of your analysis, you were treating them each as independent observations, probably because you need, pretty large sample size. But I was curious that of whether, treating it as a time series or longitudinal, you could, get information about change, like change over time. I think you were looking at body condition score as kind of one of the outcomes you're trying to predict. And I was thinking, yeah, you can treat them as independent, but then of course the cost of that is that you lose the information about development, like if you're collecting from the same animal, right? So if you're thinking about eventual biology, I was like, oh, maybe there's information in the path, right, that you're losing by doing it that way. Like maybe there's some point of transition that's important in terms of the eventual outcome.
Yeah. Absolutely, I agree with you, and we know right now that we are not harvesting the potential or the time effect, right? And for the cows, for these, I guess will be the most important component is how the cow is changing, how the cows are changing over time. These will drive the disease later on. So treating as a time series is definitely key. The problem for us was that we only have 3 time points. And so for some of these models or, convolutional network algorithms that we are using, these would not, we tried and didn't bring benefits. And the reason was because the series was too short. So now we have 60 time points before calving. And then we are using things like long short-term memory or, this recurrent neural network thing, because now this series allow us to have a better pattern to explore. And so hopefully this will be one of the things that would improve the performance of the models, is having these longer time series and looking to more the overall pattern of change, which will reflect on the cow. But yeah, you're completely right. We unfortunately we are losing that right now, but we plan to incorporate.
Yeah, it's tricky.
Other questions? Looks like Wang asked a follow-up one on since there is a quick development in deep learning architectures for computer vision transformers, but I observed that you achieve amazing results with not necessarily the newest models. Are the newest models or not the best performing ones for real life experiments? So I guess I'm maybe more of a comment, or Wang, do you want to explain that question or comment in more detail? I guess I didn't totally understand it in the chat there.
I think if you want, I can start trying to answer. If not,
yep, go ahead.
Completely right. They can ask more. So yes, so visual transformers, it's like super hot topic, very important, especially large language models are using a lot of transformers, and so visual transformers, it's being used. There are things about that we need to consider. We would need large data sets, more than we have to train and achieve performance as the ones you've seen. there are papers showing that if you fix everything as the visual transformers with convolutional neural nets, you would achieve similar performance. So, you know, there is always these sometimes strained or new models that will always come. In our case, we try to balance between exploring the newest ones and try to keep focus on the problem, the biological problem we have. And so only use if really necessary in terms of, okay, I'm not achieving good performance here at all. Should I move towards a more complex or, you know, data hungry or something like these models? And so, yeah, we haven't explored visual transformers. We have explored visual transformers from foundational models perspective. These more pre-trained models for general feature extractions and things like these we have explored, and actually they are great. There are recent models from Meta like Dino V2 or Segment Anything for segmentation. So there are these foundational models that definitely will help our field, so we don't have to retrain or train for small tasks, and they may be able to achieve very good things. But we have not trained vision transformers from, you know, with large data sets aiming to tackle a specific problem on feature extraction, which may at some point be the case if these more general pre-trained models do not produce the features that we need for our predictions. So for example, Dino V2 can convert images from RGB to depth. And so this is one way to prove, among the other things it can do, this is one way to prove that the features extracted from these models are general enough that you could do that. And so in theory, these could serve us as it is, but may not, because our particular type of data and problems are very specific, and we may need to have to train something like big with all depth image from pigs and all that at some point. But I think it's a good comment and point. We haven't explored more clearly answering, but the reasons are the ones I mentioned. I don't know if I answered that.
Looks like a little follow-up here. What are the best features you decide to use for training a model, and which features you decide to be useless?
Yeah, I think this is very problem specific. I think if you are talking about the CAL mobilization, we basically use all the features that are in the density layer, which is 1024 features for exception, which is the network we use it. And so there are several reasons that's why we did all these, and not all, no, I mean, some of them are not analytical justified. So let's put it this way. So we start training the body condition score classifier using exception was the network that performed the best compared to VGG16 on the network. So we use exception. So we had this network trained. So what we did then, we said, okay, let's get a depth image. Let's predict body condition score, but let's ignore, we don't want to use body condition score. Let's go to the density layer and let's get all the features extracted after the convolution process, or by passing the image through the network. And so we use that, and then we achieve good accuracy there. But then we didn't filter from these 1024 features, which one is the most informative and so on and so forth. We didn't do that. We use all of them. And at that point, we're not interested in what they mean, because most of them are abstract features. I mean, they will not tell us a lot about, oh, this is related to the pin bone, that is. So because we're not extracting any biological information, any meaning from the features, we end up not selecting them, which we could use the most informative features, but we didn't in that case.
Great. Other questions from the group? I have a quick one myself. So I'm working the soils area, and we've combined a lot of near infrared visible with XRF analysis.
Mm-hmm.
And they also have like a handheld gun that you can use. Have you considered using that for some of your nutritional value to get some of the heavier elements, and maybe just another data stream in there to have some more data for your training? So
So the handheld you mentioned is a hyperspectral handheld.
No, X-ray fluorescence.
X-ray for no, I haven't worked with these ones, so that would be nice to try.
That's just something we do in soil sciences, combine visible near infrared and X-ray analysis, and
Okay.
that's been helpful for getting a wider range of predictions. Like I said, more maybe some of the heavier elements on the nutritional side might be useful there. So
Very nice. No, that'd be good to look into that. I haven't worked with those. Yeah.
I guess another question is, getting it back to the NAPDC and the framework. I guess, are there any things that if there was an archive or this type of framework that could have been very useful for doing some of your studies? I'm thinking like, an image warehouse for training or anything, or there's any other things in the in this framework they're proposing that could be useful for your own research?
Yeah, this is a very good question. Yeah, I guess initially, I think access to GPUs was something that we needed. So the university is supporting us a lot with this. So they have a big center for high throughput computing here, but then our applications have to be more intensive, and so we cannot allocate the analysis of things that we want to allocate. We cannot let the farm analysis run in this at the CHC here because there are other people using it, so they cannot allocate GPUs for us to keep running forever. So we have to buy our own servers and deploy these things. And if you want to keep the farm running nonstop or things like that. This is not public though, it's not a resource that I would need, but it's just one of the constraints we face initially. One thing we developed through the AD2PI, the Genome to Phenome initiatives, Jennifer is aware of this grant. So we have a very small seed grant to develop an interface for we call this prototyping research. So basically it's, we put all the code to train, for example, image classifier in the backend, and then you would upload your image and train using any model that you want, like exception, inception, all these things, but just clicking and making these super fast for what are we saying, prototyping. So these would avoid, a lot of exploration. The exploration would be much faster. That's what I want to say. If you find something interesting, it's okay, I go this direction. So then you need to parallelize analysis, tune hyperparameters, do all what you need to do in reporting the papers and all these. But for the initial tests, you'd have some sort of like a prototype tool that would speed up exploration in image classification, segmentation, detection, and from that point, you'd take back and develop more research on it. So we started developing these. We couldn't find tools like that as we started exploring that. Again, this was a seed grant, and with the money we got, we developed the prototype set here. We have these. This could run in any GPU cluster of anybody else. And Cyverse, for example, was the one that we are talking to see if we can plug that in there. But I think this would benefit a lot of people work in that space because it saves a lot of time. Sometimes the student will write the code, prepare everything, and they see that, oh, they may not classify well, or we have problems. And so these would bring people that are not using the tool, and these would probably help people that's it's developing things and would save time for others. There's actually as a need for us too. We would use that to explore things very quick and then throw away or say, oh, we need to look at these in more details and then come back. What else? In terms of resource, I don't know, data repositories. I think this is always great. We have paid for more and more data storage. So yeah, I don't know if that's things you're expecting to hear from me, but
yeah, no, it seems like it's some great case study in the animal science world on how a framework like this could be very beneficial, to you starting out, or now you've developed some of these tools to other groups that might be interested. I know the next upcoming webinars will also be around a similar topic, so there could be some good, exchange of ideas and other things coming up. So guess stay tuned. So with that, are there any final questions or comments from Jennifer?
I just had a question, and this was more targeted more towards adoption, less from the research angle, but from the adoption among the industry side, which is that you have this really cool system that takes advantage of the computer vision side to extract important information from animals that could be useful in a dairy production system. And the question is, how do we enable the production systems to actually adopt this kind of technology? I think larger operations is probably a little bit easier. They might have more resources to integrate, things like imaging and compute into their operations. But I'm always curious about the translation to production systems, right? That, okay, maybe once you have a pipeline set up that can do good identification of animals from images, you could then, I don't know, provide that as a technology that producers could adopt.
Yeah.
And then it draws in the issues of wireless connectivity, ability to kind of power those kind of systems. So I'm just curious about your experience of trying to bridge that gap, to go from a research system that's really aimed at a production problem, like these are real production issues, to adoption, by actual producers. So, and any comment in the space in which you're working, are there any challenges there? Or
Yeah, that's a very good question, and it's very common question too. And I will give you my opinion or my view of this. That what happened is, when you think about translating in other areas, like let's think about genetics or nutrition or reaper, right? There are a lot of mature companies already commercializing something that is similar to whatever is being developed now. And so the translation is in a click almost, right? So someone develop a new protocol for reproduction. Oh, how is the translation from that protocol to implementation on the farm? Well, extension agents will take that, the research will give talks, one company will get these or license or develop the same or whatever they're gonna do, and then they have a product with some return on investment or some value that will be delivered to the farmer. So if we think about the translation from these areas, it's very clear how that happened, right? So when we go to this type of research, it's all new in all aspects. It's new in terms of how many factories do we have doing these precision ag or precision livestock. We see all the hirings, and they are pretty much all my age. I mean, since like 1918, the positions, the faculty position, like in Nebraska, you guys hired, and Cornell is hiring now, and Illinois. So the same thing is some sort in the industry for animals. Plants probably more advanced, but for animals, how many mature companies we have with products like that in the pipeline are commercializing. And so if not, the translation, it's not at the same speed as in other areas. And what I see, it's a lot of startup companies trying to mature and to get going in this commercialization path from AI or sensing technology nowadays. There are a few companies mature. I'm specifically talking more about the computer vision side for animals. So if you look at the spectrum, there are more startup companies than mature companies try to commercialize.
Yeah.
So then if you think about the translation, you see a little bit of gap between, okay, you have this research ready, but maybe the commercial space is not okay. Can it then take these and have like this pack into a product that be commercialized? And so yeah, I see that difference in the speed of the translation from other areas to our areas. Is like sometimes the researcher will have a startup company, and myself, João, may take these and bring as a product that will be implemented and used. Most of the time, the industry will do that. We're gonna do the research. Maybe there is a license or something that needs to be worked with the university. If not, okay, here's a product. Someone say, okay, let's create value on these. The pricing is the business model is that, and then they take these and bring it to the field. Definitely, saying right, speaking. Especially for most of these components that have a hardware component, is not it's just knowledge based. No,
right.
Sorry.
Bless you.
You have this hardware component that need to be sold, and so if the farmer is spending money, they want to know, okay, I paying these X cents per cow, how much I'm gonna get? So all this return on investment and economics about
Right.
the technology is important for the full adoption or for a correct adoption or progressive adoption, if you think like that. So from my experience, what I see is this. I see a lot of interest from companies that are mature, but they don't have technology, that they are looking, scouting for something.
Yeah.
Some other companies that are developing tools, but they are still in the place of, okay, I have this tool, I need to make this happen as a product,
Mhm.
and then I'll be able to take, other tools on board and so on. So that's my view for the translation. I think we need that the maturity from all the aspects of, especially from the companies, which most of the time are the ones taking the research, converting to a product, and bringing this
Right.
to the
Right.
field.
Okay. Yeah,
Yeah.
that's super helpful. And then, God, there was something else I wrote down. See, I'm making little notes here. It was, oh yeah. So the question of, how to, you were mentioning about repositories and how to store and share this kind of data. We always run into this problem, which is like, all this imaging data. What you're saying, you have terabytes of data. It's the same in the drone space, right? They're just collecting a large amounts of data, and it's difficult to share that volume of data, no matter how you slice it. It's hard to do.
Yeah.
And so I wonder about, trying to make that process of sharing a little easier. Like it's always difficult. There's this debate over, if I keep the raw data, then I give other people the opportunity to, as you said, answer questions that I wasn't thinking of when I collected the data, right? There's always the idea of, if I go back, I can use it in a different way. But the flip side is, keeping all that raw data can be very expensive, because there's a lot of it. And so there's always arguments, well, once you extract the traits from the images, then just keep the traits, right? Don't keep the raw images, just keep whatever you've extracted. And I wonder, I think in this space where you're working, there might be actual considerable value in keeping the raw data. But I'm just curious about, on that spectrum, you get different answers from different communities. I'm always curious, like, what you think about, is there real value in keeping all the raw data, given that could be hundreds of terabytes, versus trying to just store some intermediate so that, it's easier to share and it's not quite as expensive or resource intensive to work with. So what you think about that?
Yes, I think in our case, we see clear value of keeping the raw data. For several reasons. Sometimes it's not even related to agriculture. Sometimes you just need a huge data set to train a self-supervised or prove something else that is not related to the data, the domain that you have in the data per se. What I do think, and we have played with this minimally, we haven't done a lot of research on that yet, but what happened in our data sets, in our case, we have a lot of, and it probably in crops will happen the same, I assume, is a lot of redundancy, right? So you have things that are the same or very similar. And these, if you have large data sets that you have some level of redundancy, these should be somehow eliminated because it doesn't contribute to anything, training models later on, right? And so in our case, if you have cameras monitoring cows, the animals will not move in a millisecond base, right? So then this frame now will be very similar to frame in a minute. And so in some circumstances and case, I think we should be able to optimize this to keep what is relevant. The problem is how you automatize or how you automate that process, because you cannot do this by hand. You can write.
Yeah, it would take forever, right?
Yeah,
right.
So,
yeah,
so
okay.
we have played, for example, in a particular example, you want to identify behavior of cows, right? And then you collect thousands of videos, and the idea is, how can we pass this through a neural network, plot the latent space, and then just get feature whatever is very close in this space, we eliminate and we subsample from that space. And now I'm getting rid of all these redundancy in that particular thing. Or For example, let's say I don't have enough image of animals drinking. How can
Right.
we identify some features that we are interested in this whole data set that is underrepresented automatically and say,
Mhm.
okay, I don't need more of this image, I need more of this type of image, right? So
Mm,
what I think
okay.
that would be interesting from keeping all this data in our case is, okay, I'm keeping all this huge data set, but whatever is redundant, we don't need.
Right.
Right.
Right. So yeah, well, I get we do have the same issue in other application areas like plant sciences, where the background is the biggest part of your image. So when you do the segmentation and you pull
Yeah.
out what you're really interested in, the
Yeah.
vast majority of the image is background, which
Yeah.
you don't really care about. And so
Yeah.
the idea would be, can you then extract what's important from the image and then maybe just store a generic background, which represents the background for all of your all of your images, right? So if you could reconstruct pretty easily. Yeah. Okay.
Yeah, or
Okay.
just you can just save the chains frame of the frame and not the whole frame.
Yes.
Things like this. Yeah, but in our case, when you think about time, you collect things over time. In our case, for plants, they will stay forever in the plot. They won't move around. But the animals, no, the
I
animals
hope
will
not.
move.
I hope not.
Yeah, I hope
Start
not.
walking around. We got
Yeah.
another problem. Yeah.
Right.
Uh-huh.
But for the animals that we are tracking them, some of the frames will be similar because it's second 1, second 2, second 3, and everything
Yeah.
looks the same. And we are hopefully trying to find a drinking example, and drinking happen
Yeah.
minimally in few minutes and a day, and this sometimes is what. So if we have the behaviors well represented and distributed, but not with a high level of redundancy for a behavior type of data set, for example, I think maybe, I don't know.
Right.
In our case, it doesn't add anything, unless you have a particular justification to have every single frame. But other than that,
Gotcha. Super helpful. Yeah. Okay. Thank you very much, Joel. That's good information.
All right. Any final questions? I know we've had some people drop off and getting pretty close to the time here, so. All right. Well, behalf of the committee, we greatly appreciate it, Joao, you joining us, and look forward to you participating in future webinars and workshops and the whole project. So, all
thank you
right.
very much. No, the pleasure. I appreciate the invitation.
Great.
Thank you very much, Joel.
Thank you.
See everyone
Thanks, Trenton.
in a month from now. So, all right. Take care.
Bye.
Thank you all.